in cell biology and tumor research. The cell scratch assay is a fundamental and crucial experimental method. However, the traditional observation method has obvious limitations. It requires taking samples at fixed times for observation and photography under a microscope. It is likely to contaminate the samples and easy to forget key time points. The MSHOT live cell imaging system can solve these problems well, adopting the inverted microscope principle and compact design. It can be placed in an incubator for long-term cell observation without opening, supporting automatic time shooting, intelligent analysis, and remote monitoring. With parameters set, it enables continuous image capture, scratch area tracking, healing area change analysis, and export of charts or videos, ensuring no moment of cell dynamics is missed.